Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show, bringing you another really great guest today, helping to create a better tomorrow on some really unique fronts. Today we have the honor of being joined by Dr. Dana Merriman, who is Professor Emerita in Biology, University of Wisconsin-Oshkosh. She's also the Squirrel Colony Director and Adjunct Professor of Ophthalmology and Visual Sciences, Medical College of Wisconsin. With her bachelor's in biological sciences and her PhD in physiology and cell biology, both from University of California, Santa Barbara, as well as having spent time as a postdoc fellow, University of Utah Health Sciences Center, a core focus of Dr. Merriman's laboratory research over the years has been the development of this fascinating captive breeding colony of 13-line ground squirrels. And this is a really unique model. It is one-of-a-kind breeding program, and due to this species' really unique visual system, which is extremely cone dominant, they are a diurnal species, as well as their really impressive physiological ability to survive hibernation for long periods of time, up to 6 months without food and water. They have served investigators with a really important capability with both animals and tissues from these animals, United States, Asia, Europe, for the last couple of decades. They've also been very core to Dr. Merriman's own work in vision research, including cone cell biology, retinal function, during the metabolic state transitions that are associated in hibernation. Over the years, Dr. Merriman has expanded her research horizon outside of vision into neurosciences. And in recent years, she's collaborated on studies and focused on muscle physiology, viral genomics, molecular biology of transposable elements, and interestingly, the comparative genetics of coat control patterning. Her lab's research has been funded by various sources over the years, including the National Institutes of Health, the National Science Foundation, the Foundation for Fighting Blindness, as well as University of Wisconsin System Comprehensive Campuses Tech Transfer Organization, WSIS. We're lucky to have her with us today. Dr. Dana Merriman, thank you so much for taking the time to come on the show.
Thank you, Ira.
It's a really, as we were chatting beforehand, and I mean, it's a really fascinating area of biology, and I'm excited to have you with us today. Before you get into anything, though, I would love to start off just by giving you the floor for a couple minutes to talk a little bit more about your background. If you can take us a little bit to the early days about, in essence, who you are, where you grew up, and how you first got interested in biology in general. I think that'd be a great way to start stuff off today.
Sure. Well, I grew up in Iowa City, Iowa, as a town kid, but had big connections to the Amish community and their traditional ways of life, which included a lot of livestock. And I was reading zoology books very early on and always trying to convince my mother to let us have more and more pets. And so I was really hooked on the wide range of zoology fields, and from a very young age. And so it was really clear that once I got to college, it would be a biology degree of some kind. And animal behavior in particular always fascinated me. And that, of course, segued to my neuroscience training for my PhD postdoc and since then. When I was 15, we moved to California and sort of saw the ocean for the first time in person. But as a kid in Iowa, we weren't allowed to watch too much TV. We were allowed to watch Jacques Cousteau's Undersea World special. And so I immediately got fascinated by the organisms of the intertidal, especially on the California coast. And so when I finally wound up at UC Santa Barbara for my undergraduate degree, it's a fabulous campus right on the coastline, and just going running and walking, you see a lot of organisms wash up or in their natural habitat. And I took a very broad biological sciences bachelor's degree, which maybe reflects my lack of focus, but also just broad interests, which thank God have served me well to this particular day and the work I do now. I had a year of invertebrate zoology with Dr. Demris Davenport, who was a wonderful naturalist, trained at Harvard, who he and his wife personally took us on field trips to the intertidal for our class. And then I had my first research experience in our third quarter of doing a behavioral study of snapping shrimp under the direction of James Case, who was another oceanographer, neuroscientist at Santa Barbara. And so I got exposed to a lot of different kinds of biology and took a neurobiology class from the man who would become my PhD advisor, Stephen Fisher, who is now retired, but still quite active in research. And we just published a paper together about a year and a half ago. And so it's been UC Santa Barbara was a fabulous education and an incubator for my interests. And I also, I should mention that I was a poor kid on scholarships and work study. And so my first year at UCSB as an undergrad, I got a work study job in the oceanographic laboratory of Bruce Robeson, who's senior scientist at Monterey Bay Aquarium Research Institute. And went out on oceanographic cruises with him and the rest of his team as an assistant, collecting specimens from the pelagic and bathypelagic zones. And so that was my first exposure, now that I think back on it, to extreme physiology, animals which have evolved ways to combat the challenges to which they're put in the ocean. And that experience, plus Steve Fisher and my PhD advisor, he always emphasized, know the biology of your model species. And I think in my PhD, I ended up working on 5 different species overall. He also had an agreement with the local Santa Barbara Zoo that if an animal passed away, we would be able to have the eyes. And so we had this library of exotic eyeballs. And I was, trained from the beginning, really to think broadly about organisms and the August Krogh principle, which is for every physiological problem, there's an ideal species in which to study it. It just became part of my personality, I guess, as a scientist. So when I was in my doctoral education, it was sort of the 80s, and the golden age of molecular biology was dawning, and a lot of people I knew were cloning genes and doing a lot of molecular biology. And I confess that I was a physiologist and a cell biologist and a closet zoologist, animal behaviorist throughout all that. And so if I had one regret, it was that I didn't take anything beyond an introductory molecular biology course, but I loved it. And I actually sat through it a couple of times because the university started a note-taking service for undergraduates. And so again, trying to make a buck wherever I could, I would go take notes in classes ranging from molecular biology to genetics to developmental biology and even meteorology. They hired me for that class twice. So yeah, I'm kind of a jack of all trades scientifically was my pedigree. In my postdoc, I chose that one in part because it was a program that was just getting funded by a donor, John Moran, who built the Moran Eye Institute in Salt Lake City there at the University of Utah campus. and had a dynamic postdoc advisor, but I was able to actually introduce myself in that postdoc to a brand new system to me, which was teleost fish retina, famed at the time because it had been discovered by others, that there's a rim of persistent embryonic stem cells in the retinas of teleost fish, and some cool work had been done using a neurotoxin to destroy the retina and have it grow right back. And so I was really interested in natural regeneration. Mm-hmm. And so with Eric Lassiter and then also the fabulous ultrastructurist Helga Kolb at University of Utah, I was able to satisfy some questions I had about fish retina and think more broadly again about models. It was harder, and I think it still is, to get funding for some of these non-traditional animal models, and as mouse and later rat mutants that you could genetically engineer came about, they really, sucked a lot of the scientific oxygen out of the room. But and I did work very successfully in my early years at Oshkosh on rat mutants, mm-hmm. With Steve Fleisler, now at Buffalo, Dan Organiziak, retired from Wright State University, fabulous colleagues, wonderful scientists, using both pharmacological and genetically modified rat models to model human diseases and conditions. Light damage, for example, is Dan Organiziak is Mr. Light Damage. And so, I really had a lot of luck in being in the right place at the right time to learn various animal models, their pluses, their minuses, and then the realities of trying to get funding if you just had your heart set on one that wasn't a rat or a mouse. Yep. In the last 20 years.
Yeah.
I don't denigrate those models at all. They're incredibly useful. But as I became more independent, I started thinking about the mismatch of nocturnal rodents, which are cheap and easy, I get it, mm-hmm. with the human fovea, which is what everyone in the audience is using to look at my face or your face, or to read something, or when you're driving to look at signs. Your fovea is this 1 millimeter spot of retina in each eye that you use for all your high acuity vision, and it's 100% cones in the human being. And so there's just not a similar retinal anatomy in rat or mouse. There's also differences in the size of the lens, in the amount of visual cortex allocated to visual input, and therefore the amount of the brain of these mm-hmm. animals allocated to visual input. And so while the primate model is unmatched for human retinal cell problems, it's difficult to work with. They're expensive. They can be hazardous. There's a lot of ethical concerns. Rodents remain sort of a go-to guy for retinal cell biology. Although I should say that organoids and cell culture systems, they're making inroads, and we've been involved with that, building iPSC lines from ground squirrel and retinal organoids, and some other organoid tissues outside of the retina have been done by clients of mine in the past. And so, but the 13-line ground squirrel caught my attention in the early 2000s because it was close by and it was a hibernator. And my PhD lab, Steve Fisher's lab, had also shown in 2002 that when you do a retinal injury, an experimental retinal injury called experimental retinal detachment, mm-hmm. which you can model in a lot of animals to really closely approximate what happens in traumatic or systemic human retinal detachments that happen for a variety of different reasons. The animal models are really robust, and Steve Fisher's devoted his career to exploring different models. When they tried California ground squirrel for the first time, what they noticed was that while detachment did harm the photoreceptors based on their separation from the blood supply at the back of the eye. The follow-on sequel scarring and neurogenesis attempts at reestablishing synapses, which corrupts the wiring diagram, didn't happen in squirrel nearly to the extent that it does in mouse or cat or human or primate, what have you. And so that rang a bell when I read that paper of this inborn resilience, which may, in some people's mind, limit the squirrel model toward its applicability to human retinal detachment. But to me, it was, wait a minute, evolution solves some problems in this species that create unfavorable environments for, say, stem cell transplants to fix a detached retina, to try to put cone cells back into the damaged eye through using programmed induced pluripotent stem cell. You know, these animals don't have that follow-on damage that makes that an unfavorable environment. How did that happen? What are those evolved resilience factors? And so I was hooked. I was hooked. And I was very fortunate at that point to be in Wisconsin, because just down the road from me was at UW-Madison, Hannah Carey, who is a giant in the field of hibernation physiology, had an established 13-line ground squirrel research program. In their logistics, they would catch wild ones every year and raise the litters in captivity, and that's how they supplied their animals, because the received wisdom 20 years ago was you can't breed this species in captivity, and they're hard to handle. Well, it's true. If you go back into the literature of the 1930s, there was a lot of 13-line ground squirrel work done during the Depression, I think because they were cheap and easy. People had tried to breed them in captivity, and there's one paper in 1933, I forget who the first author is, I think it might be Johnson, but a group had put something like 600 animals into a pen with, you know, dirt and grass and fencing underground so they couldn't ingress or egress, and tried to breed them and got 0 litters. And so that augured poorly for any attempt, you know, to try to create an outdoor environment as a breeding colony. But in 1988, Holmes and, I can't remember the other person's name. It'll come to me. They had done a very small scale captive breeding and published on that. I think it was something like 8 females and 8 litters, which gave me hope. And so I did my best our first year. I visited Hannah, I think it was in 2002. She was able to supply me with eyes from animals she was euthanizing for other reasons. And that gave me my entree into their visual system. And then I trapped my first animals, and we raised our first litter, captive bred litter, the next year. And it just kind of went from there. Our first litter was born in 2003, and it was 2007 when we had enough production that we were able to start supplying our first client, Wei Li, who's the director of retinal physiology at the National Eye Institute, and our client base has grown ever since.
It's so interesting, because before the show, Dana, I was spending some time in PubMed and going through a lot of these, a lot of this path that you were just talking about in the publications went along with them. And, as you were saying, you see rats, you see rabbits, then the ray fishes, the teleos come along. You did a paper on leeches, I believe, as well.
Yes.
Come 2006, you published this seminal paper, Capture, Care, and Captive Breeding, 13-line ground squirrels. And I want to go. We're going to go in 2 directions. We're going to get into sort of the cryptobiology, the hibernation physiology, and so forth. But before we do that, any interesting stories per that 2006 timeframe about the breeding events? Anything unique or weird, or you want to highlight before we get into the the genetics and the deeper
Sure.
biology?
Well, and 2006 was such early days, and I've learned so much since then. So it'll be hard for me to regain the mindset of the beginning, because that's not where I am today.
Sure.
I can tell you that until about 2 weeks ago, my lab was entirely staffed by undergraduate students who were first volunteers back then, and then came aboard as NSF REU scholars, McNair scholars, paid. Once we started getting an income, I was able to pay my staff long before years and years, a decade before I paid myself. And so I want to shout out to them, because they made all of it happen. And I'm still in touch with almost all of them back from then. But we knew that they were seasonal breeders. We were able to create a hibernation environment in a cold room. And I started from the beginning. I wanted to recapitulate the animal's natural environment in, lab husbandry setting with cages and clean rooms and all that stuff that you have to do to be able to conduct animal research in our welfare environment, which I completely agree with. The point being that was fortunate, because we hibernated them and then brought them out and then put boys and girls together in cages and just kind of waited to see what would happen in rooms in which it was typical standard lighting. And that's something I'd really like to experiment with, is changing the color temperature of light bulbs away from standard fluorescent, just to see if that makes a difference. But we had day lengths that increased and decreased based on ambient. So a lot of lab animals are kept on 12 hours dark, 12 hours light, which is not anything that a 13-line ground squirrel usually sees, because if you look at when they come out of hibernation and go in Wisconsin, 12-hour days are just starting at the equinox. And so it'd be a rare ground squirrel in the wild in Wisconsin who would see 12-12. And so that, I think, was really part of our success, is that we gave that Zeitgeber of day length right off the bat. And then we've always tried to feed them enrichment treats, is what you kind of think of it. But live insects, worms, crickets, they love those. Turns out that scrambled eggs, because they eat a lot of eggs during they're in Wisconsin, next to raccoons, they're the second biggest predator of ground nesting birds. And I should mention feral cats are at the top. Feral cats, but in wild animals, raccoons and 13-line ground squirrels prey on the eggs and the nestlings of ground nesting birds in Wisconsin. And so scrambled eggs was something we had to cook them, so they're cooked, but eggs they like, and they do really well on those. We also played around. I wasn't sure that rodent chow was the right thing to be feeding them. And I'm not a nutritionist, I'm not, but they're omnivores, and they're predators in the wild. They kill and eat mice, snakes, obviously insects, obviously birds, each other if they can get a hold of each other's young. They've even been documented eating roadkill. And so I thought to myself, these aren't alfalfa pellet animals. These are like dogs or cats. They eat meat. And so we started them on a carnivore diet. Cat food in 2006, but I think by 2012 we had moved to dog food, which we've experimented with a couple of different diets, and skin issues in the laboratory setting recommends one particular brand name diet over others that we've experimented with. But the point is that we got lucky early on, I think, by paying attention, as Steve Fisher in my PhD program, know the biology of your model organism. And while just putting a male and a female together isn't relevant to their real lifetime, because they escape tunnels and so on. And we give them tunnels, but it's not the same as being in several acres of territory where the female can say, go away. fact of the matter is, once a female is bred, it's been well established that within hours, there's not only a copulatory plug that kind of prevents further matings, but within hours, something happens to her pheromones and she ceases to be attractive to other males. And so, you know, if you observe copulation, then you know she's bred. And subsequently in the last, since 2006, how many years has that been? 12 years. We've figured out that pretty much as long as there's a positive sperm count, you do a smear, right, post-copulatory smear. If you find sperm and there's a plug, she's pregnant almost 99% of the time. So we just sort of put them together to let them do their thing. And these days, however, we put them together daily under observation by a trained observer to see if he's showing any interest. And we also know that there are certain preludes to behavioral estrus. And so there's no point in putting them together until she's ready. She controls it.
Gotcha.
And so, but back then we just put them together for 25 days and then separated them. The gestation's about 27 days. So we would leave them together for 25, separate them, and were there any babies? Mating copulations in the wild are spread, typically they cluster over, depending on the weather we found, because Wisconsin weather has been very crazy the last 5 years. Depending on weather conditions, you can see in a particular single habitat in wild ones, copulation has occurred in just a 2-week time frame. The births are all clustered, meaning the copulations were all clustered. We don't get that kind of tight clustering in the colony, I think, because they're not as circumscribed by weather events. It's always nice in the colony. It's 70 degrees and there's plenty of food and there's no predators, and you know, but within about a month time frame, about 30 days. And so yeah, but we got enough litters that we could supply people, just putting them together and seeing what happens.
Yeah.
But in the last few years, because of demand for time pregnant females and for embryos of certain stages, because once an animal model comes into play, the embryonic development becomes an issue, especially if you're researching diseases or conditions that might arise prenatally. And so we had to really observe copulations to nail down the gestational day. We've also repeated work done at UW-Madison as a PhD thesis during the Depression. That's Foster, 1934, great paper for the tools at the time of how soon after copulation do you start seeing 1 cell, 2 cell, 4 cell, 8 cell, all the pre-implantation embryonic stages. And those are important to some work we're funded to do by the National Eye Institute now on creating a germline transgenic 13-line ground squirrel, because you need to collect those pre-implantation embryos and gene edit them in vitro and then put them in a recipient. And so all that really detailed copulation timing and study of their reproduction in captivity has really just been the last 4 years, but we've learned a lot. And yeah, so I think that answers your question.
Yeah, no, that's fine. It's you know, I don't normally, you know, think about 13-line ground squirrel copulation dynamics, but no, that's cool. I mean, obviously you got to think about all these things. Yeah, I mean, obviously, you know, there's, as I said, there's two. You know, as you've been saying, there's two roads here. You need the model. You create a unique model. You got to be able to provide the model and have it, and then obviously continue with your own research. And you know, then as you were, you know, saying in the intro, you know, you were introducing us to this really complex resilience, robustness per the remodeling of the eye. But you've published extensively on a variety of other really cool, I'll call them superhero type capabilities, in terms of their ability to do really neat things with exercise endurance pathways while they're lying there for 6 months so they don't, waste away. You've published on their ability to keep cold sensitivity at bay during this process, and then the brain, this unique ability to alter the excitability of their neurons, and we'll get into a little bit later probably about during those 6 months the brain also changes a lot. Give us a little, because it's more than just the eye, and I really I want the people to understand how sort of a little bit more about some of these superhero capabilities and why this is such an important model.
Sure. So again, using Wisconsin as the context, they will come out of hibernation where they've been sealed underground in a dark soil temperature. And by then it's about 40 degrees F. It's like your refrigerator at home. They've been sealed in a chamber without food, without water, without a heat source all winter, going through cycles of torpor, which is low body temperature, low metabolic rate, and a very minimal motor program, which says stay in the fetal position, dorsal upright with respect to gravity.
Yep.
And in the midst of torpor, you can go in the refrigerator and almost unroll them and watch that motor program slowly recapitulate that fetal position. And that's about all the arousal you'll get if you keep them cold, dark, quiet, and you only handle them once. And so the nervous system is barely functional. These animals are not conscious, but they arouse on a circannual cycle, we think. We don't exactly know what triggers it. In Arctic ground squirrel, there's fascinating data suggesting that somehow snow cover is a predictor of arousal timing, given the variations in when spring comes in the Arctic. Those animals have a very short time frame to reproduce and prepare, fatten for hibernation. Our guys have another month at least. But in Wisconsin, April, they dig their way out, so they can't have lost any bone stability or muscle because they've got to dig out a tunnel they've sealed behind them to prevent anybody from coming and eating them while they're helpless. They have to be able to get through ground that may be partially frozen. They're above ground and they've got a very limited food supply. The birds haven't laid eggs. There aren't insects yet. And so they have to be able to navigate a relatively nutrient poor world on arousal. The males come out before the females, probably because they need time to regenerate their testes, because they don't carry full testicular size and function and anatomy during hibernation. The testes regress after the breeding season. So the boys come out, they have to regrow their reproductive apparatus. The girls come out, and the previous fall, their reproductive apparatus has actually started to prepare for ovulation, but it's paused when they go into torpor, into hibernation for the winter. So the boys come out and you see them foraging and trying to get food and so on, and also scout the tunnels of the girls, because the girls, when they show up, some of them in the colony will actually be reproductively receptive, behaviorally estrus, within 2 days of coming out of hibernation. Most of them, it's longer. It's more like 7 on average in the colony. But in the wild, a male has to be ready because they are promiscuous. They have very large testes for the size of the animal, which evolves in promiscuous species. The males actually will run the territory, and this is not my work. This is work of wonderful field biologists like Schwagmeyer, for example. And they will actually note sort of where are the females' home tunnels, And the males will run those tunnels, and if the female's estrus, he'll chase her. He'll try to mate with her in that springtime. Again, this will be in mid-April at the earliest, usually in our part of Wisconsin. Farther south, it might be a week earlier. Farther north, it might be a week later. Because I work with field populations that span Wisconsin, which is a pretty tall state. And so we find these latitudinal differences that are then further influenced by weather. As females are mated, they drop off the map and he doesn't check those tunnels anymore. It's really wonderful. If you're going to be promiscuous and have these large territories, it's a great way to sow your oats in the most efficient manner possible. But the females have had a challenge because they've been starving all winter, and they come out of hibernation with very little body fat, and they have to rev up their reproductive apparatus. They've got to gain enough fat, to sort of make that permissive. Then they've got to start gestating these embryos, and gestation's like about 27 days, 26 days sometimes. So within a month, they're giving birth. Then they're lactating. So I think the female 13-line ground squirrel has more superhero powers maybe than the male in some ways, because she lactates, and then she's got to fatten her own body to be ready to go into hibernation in fall. So what often happens in the colony, and is somewhat mirrored in the wild, is they've come out of hibernation in April, breeding in May. If you're a female, lactating in June. You've got July to get fat as a female. The boys have been able to fatten in June and July. The boys will actually start hibernating in August. There's a sex split both in timing of arousal from hibernation in the spring and emergence into hibernation in the fall. And we see this play out in the colony as well under these very stable environmental conditions where the day lengths are getting shorter. And I'd love for someone to crack the code of the circannual cycle in these animals, but to my knowledge, no one's really sure where that circannual clock is. There was a paper providing evidence some years ago that it's not 365 turns of the circadian clock, that it's something else. But so April, May, June, July, 4 months of activity for a boy, you're back in hibernation. A 5th month for the girl because she's borne the burdens of gestation and lactation, but in September, she starts hibernating. And in really great warm environments, they might not hibernate until October, and further south, it might go later, further north, it might be shorter. There's these regional latitudinal variations. But what we're seeing is an animal that has 4 to 5 months to do its reproductive duty. And then it goes into this prolonged hypometabolic state where instead of the heart beating 200 beats per minute, it's more like 11. Instead of, I don't remember what the respiratory rate is, but it varies so much based on handling and stress, but their breathing can become intermittent or very, very infrequent. One of our graduate students at UW-O did his PhD in British Columbia, and he's done a lot of wonderful, very recent work, Ryan Sprenger, on respiratory control during hibernation. So that's a nut that's very interesting to crack, because if people respired that slowly, they'd get tremendous amounts of brain damage and hypoxic organ damage, and squirrels tolerate it. I think I want to also give a shout out to Sandy Martin's lab at University of Colorado, Denver. They published a very important paper in 2011 showing that the preparation for hibernation, I've mentioned that the onset of hibernation, digging that tunnel, making that chamber, sealing yourself in, and you're gone. See you in April. That there's a whole prodrome, a metabolic prodrome that can begin in July, August, certainly manifests in the early fall months. And they've published very important data, in my mind, for those of us interested in hibernation physiology across organ systems, that a fall squirrel is not a summer squirrel, that the preparations of the organ systems for all the hardships of hibernation, the cold temperature, the low breathing rate, the low perfusion rate, being in an underground chamber where undoubtedly it's not only cold and always dark. Think of what a stress that can be on an animal by its vision in bright sunlight. Also high CO2 levels. I mean, they're barely breathing during torpor, but they're sealed in. It's like being in a space capsule. So there's all these fascinating challenges, and the preparations for those begin in late summer into the fall. And the most obvious one is they get fat, they get obese, they put on adipose tissue, but it goes much deeper than that. And so Sandy Martin's paper, I believe the first author there is Elaine Epperson. Super, super important paper, because I think some past work with hibernating species, if the animal is euthermic, if it's warm and active, eating, sleeping, doesn't matter what time of the year it is, it's classed as a physiologically aroused animal. But if this prodrome of hibernation preparation is happening, are they really the same animal they were, say, in May or June? And so you have to look at your data from animals that are proper temperature and active in the fall aren't necessarily going to match the data of fully euthermic in reproductive condition. And we're always thinking, you know, in the back of our mind of matching human biology, and we don't hibernate. We don't breed seasonally. And so when you think about a hibernator and matching human, that May, June, July, a fragment of their annual cycle is probably, you know, super important for validating that model. But the hibernation periods are valuable too, because if we need to hibernate astronauts, for example, to save on payload for manned missions to Mars, what's the safest way to do that? What can animals that survive hibernation with no problem? I think it was one of the principals of Fauna Bio in one of their interesting talks. That's the company that's really capitalizing on hibernator biology for novel pathways to drug discovery. Fauna Bio and we are, my lab have become partners recently. We've worked together in the past, the principals and I, but now we're formally partners. But they've said they have the equivalent of, I don't know, 24 heart attacks each winter, with the kind of slow flow of blood and the slow, and yet they can dig their way out and mate right away. And that's some pretty impressive resilience.
That's I mean, just, you know, thinking about everything you just went through, and whether we're talking about the heart or the brain or the eye, I mean, there's so much here in terms of, as you're saying, the obviously we don't hibernate, but nonetheless the potential translation to some of these unmet medical conditions, and you know, it gets me now think, as you mentioned again in the beginning, your work in, you know, not just, okay, we created the colony, we've studied this amazing physiology, but then several years, well, I don't know, back in 2018, I believe it was, when you first did your you started collaborating with the NIH researcher on the induced pluripotent stem cell work, which the press release at the time was all about hibernation in the petri dish. And here, you know, once again, a broad possibility where, okay, we can take these cells using and we've discussed IPS on the show in the past, we can use the Yamanaka factors or other reprogramming tools to spin these back. And now whether it's cardiomyocytes we want to create, or clone cells, or various other lineages, now you really have the ability to, as you know, as dissect these various tissues and look for whether it's the genes or the epigenetics or all the other interesting, cool things that are happening that allow this resilience, that allow this robustness. What's been going on in terms of the IPS work that you can talk about, whether it's with the NIH or the Foundation for Blindness? What are some of the cool things you're seeing in terms of hibernation in the petri dish?
I have to admit that the principle for that paper you mentioned Jingxing Wu's paper out of Wei Li's lab at the National Eye Institute. They're in charge of that.
Okay.
And Jingxing has moved house to China, and so there's been sort of a disruption in the work.
Okay.
And I can't tell you what they're working on right now. That line turned out to be a female line.
Okay.
So another group at the Lamba lab, Deepak Lamba at UC San Francisco, their shop is working on a male line of iPSCs. And I didn't know this until I started working with these people because, again, I'm not a molecular person at all, that it's useful to have a male line because if you transfer male iPSCs into a female host,
Okay.
for a transplant therapy type experiment, it's very easy to prove that the cells that have, you hope, successfully flowered and done a job are from the iPSCs because they are male.
Mm-hmm.
They have a Y chromosome, right? You can tag that. My understanding is that iPSCs are harder to handle than people imagine, and that squirrel iPSCs have presented some obstacles. Honestly, the iPSC work that's being done very successfully, and there have been some recent presentations at the Association for Research in Vision and Ophthalmology meetings, which just happened in the early May this year, out of Dan Lipinski and Joe Carroll at the Medical College of Wisconsin. Their graduate students, Jeannie Yu, presented some of this. They're doing some amazing work putting human iPSCs that have been programmed into cone pathways into damaged squirrel retinas.
That's interesting.
So that's a cross species. Right. And the advantage there is that if we get living cones out of it that boost vision, it's of course the big dream of transplant biology is to be able to put stem cells that have been derived into the cell type that's missing and let them take root and, in the case of the retina, synapse appropriately with surviving interneurons to recapitulate the circuitry in a normal fashion and reinstate vision function. If you can do that, then having human ones, it's really easy to say, yep, these are not just cones that, I don't know, migrated in from other parts of the retina, squirrel cones that did this. These are the human ones because they're so easy to pick out from the crowd because they're of human origin. So my understanding is that the cross-species iPSC work is bearing fruit, and I would just refer interested parties to look at the ARVO abstracts from this past May for what was presented there.
Okay. Yeah, I figure. I mean, it's just, as I said, it's. I enjoyed reading through all your publications over the years. And I see the direction. Obviously, we're in a unique time, as you were saying, with novel models, and there's lots of things you can do with novel models. You can obviously breed, but at the same time, all these new technologies are coming online to, once again, blast apart lineages and look at specific things. So I think it's just another neat direction that your program has taken. So I just wanted to.
And as for some of my clients, that's their clients. I supply them animals or tissue, and off they go. And I don't know all. I can't claim to know exactly what they do. For example, years ago, WISIS, the organization that you mentioned earlier, which funds a lot of pilot projects for the not R1 institutions with the University of Wisconsin system, WISIS got a bunch of biologists together in a room and said, tell us in 5 minutes, give your elevator talk about what you work on. And I talked about hibernation in squirrels. And at the end of our roundtable, a person, Scott Cooper at UW-La Crosse, came over and talked to me and said, he's a hematologist. And he said, this is very interesting because the heart's barely beating and it's all cold. I said, that's right. And so he was quite interested, and we gave him a free sample of squirrels, got him started in squirrels that year. And I'm not a co-author, and I don't understand hematology I've read his papers, but I'm not a hematologist, but I would refer the listeners to Scott Cooper's work on 13-line ground squirrels, because if I'm understanding this correctly, it's very hard to store human platelets. A lot of patients need platelets to help them clot, and we like to just give them platelets. But they're hard to store because apparently in the cold they fragment or explode or something. And of course, squirrel platelets get cold. So how does that work? And Scott's had a R15 award from NIH and went through all that, has published some great papers explaining how the various blood components of squirrels are preserved during hibernation and then redeployed on arousal. So they don't have to make platelets de novo all of a sudden from, spring when you're doing all this other stuff. And that could maybe contribute to blood banking improvements. There are so many things my clients do that I'm not intimately involved with. And so I kind of know how cool it is, but I can't claim intimate knowledge. And the stem cell stuff, from what I understand, is ongoing and being worked with, but I can't claim to know exactly what is done there. And I have to keep my lip zipped about certain things.
No, I hear you. I completely understand. Not an issue at all. As I said, I just I really wanted to profile the broad picture of I call them superheroes, but the way you have dissected all these different pathways over the years and then built the system is just extremely fascinating. It's just
Thank you. Well, and a couple of things just for me to mention is Wei Li, again at the National Eye Institute, published a really interesting paper. I think it was this year or last year. And it was an idea that I had discussed with him back in 2011, that we know that the retina, for example, doesn't undergo scarring. And retina is a piece of the brain that sits outside the skull. And when you damage the brain, you not only lose the neurons that have been injured, but there's a scar program from glial components. Glia are called the support cells of the brain, but they do way more than that. They actually do communicate with neurons and have their own private communication pathways. But glia change their gene programs and their physiology and even their location, and create these scar tissues that are persistent and can prevent regeneration of neurons. And so this has been known for a long time in the central nervous system. And what was notable in that 2002 paper on ground squirrel retinal detachment is those scar programs did not enact in California ground squirrel. And so, retina is one thing. It's a tissue that takes a beating. It's outside the skull. But I got interested in optic nerve, which is more brain-like and, of course, penetrates into the brain, into the thalamus. And so I was interested in models of optic nerve damage. And optic nerves get damaged a lot of different ways: penetrating brain injury, a tumor that might grow. There are sometimes almost like bone spurs that grow in the skull, and they can press on nerves very, very problematically and cut off vision and damage the optic nerve. And so people do these experimental optic nerve compressions, and that in all animal species except one engenders this scar response and a lot of permanent damage. But Dr. Lee and his team showed in ground squirrel that the scarring is suppressed, and they actually had, I don't remember the name of the compound, but they recapitulated this in vitro with squirrel cells, and then with the compound present with, I believe it was mouse cells in vitro, and also showed suppressed scar responses. So there's another very recent application of sort of the baby steps of drug discovery from a hibernator that's naturally suppressed problematic processes in the central nervous system. Now we try it in vitro in a mouse, a non-hibernating rodent, and then you go from there. What the future plans are for that, I don't know, but that paper is pretty promising, and it got some press as a result, and it's one of the first real translations in central nervous system from a hibernator. And of course, it's in the visual system, of particular interest to me, but it's getting back into the brain.
Right.
And so, you know, and the list goes on and on.
Right.
Everything from one of our faculty members hired after I got the squirrel colony going was Courtney Kurtz. Courtney Kurtz was a PhD student of Hannah Carey at Madison, whom I referenced as one of the leaders of hibernation physiology. Hannah recently retired. Courtney is just down the hall from me, and she's focused on sort of general metabolic. She's an immunologist and a gastroenterologist, and there's a huge immune system component to the digestive tract. And so she's really interested in microbiome, immune system, GI function, which of course shuts down during hibernation. In that fall prodrome, one of the last things the animals do is stop eating. So they go into torpor for winter with an empty gut. Which is a stressor. You know, a lot of animals, you don't feed them on a normal schedule, they develop ulcers and all kinds of problems. But hibernators do it. And so she's also more recently been funded for the whole onset of obesity and almost the diabetic-like component to that fall prodrome, which then resiliently self-corrects upon emergence from hibernation. And so her graduate student is defending her thesis on this very topic tomorrow at noon. So
that's that's what we've actually heard, not in the squirrels, of course, but when we had Tim Lasky on the show last year from Medtronic, because he's spending time with the bears, and the same deal. You know, whatever however much they consume in that period. And no diabetes, no heart disease, and
and now that we know, I mean, I've been teaching physiology to freshmen on up for 30 years, that there's sort of, I call it the square of coordinate control: the nervous system, the endocrine system, the immune system, talking to both, and now the microbiome. And from a husbandry point of view, that's become, I'm not a microbiologist, but I have forced myself to learn some microbiomics, because I believe in the laboratory setting, when you work with any species, and ours is a wild species, rats and mice are kind of a domesticated species, right? The focus on sanitation in a laboratory setting is endemic. It's systemic. It's in the guidelines, right? But are we depriving, especially our out of the wild animals, are we depriving them of a microbiome component that might give us, say, better reproductive success? And there is this concept of rewilding mice, and there have been some recent papers on that, and I'm studying those carefully to see if there's a way we can reintroduce soil. And I'm not talking soil that's been gamma irradiated, like much is when you give lab animals hay, for example. It can be sterilized hay, so it doesn't have the microbes that they would be getting. But hay and soil seem like natural inputs to a 13-line ground squirrel cage. And so I'm very interested in doing that and seeing, from a well-being standpoint and also a reproductive success standpoint, and we do pretty well with our animals, raising them and getting them pregnant and raising them. We've done it very consistently, producing about 500 pups a year for almost 10 years now. Space is our constraint, but if rewilding the microbiome provides some sort of signal that, yes, it's good timing for estrus. You know, wouldn't it be interesting if the soil components in the spring provide a go ahead and breed Zeitgeber, whereas the soil component microbiome in fall, late summer, fall says, hey, better think about fattening and going night-night. They do it on average with just the day length changes we provide them, but not all of them. It's not 100%. And if you don't hibernate in the wild, you die.
Right.
You cannot survive winter.
It's really interesting you bring up that microbiome component, because Mike Levin from up at Tufts, who I've had on the show a couple of times, he spends time with the membrane potentials and sort of the whole bioelectric theme, and he's published on Arctic ground squirrels actually, and this how, as you were saying, how all this pruning of the CNS occurs, and then voila, when they wake up, everything's cool. But he's also recently published on the microbiome and regeneration, and that when that snake comes along and bites the poor little salamander's leg off, there's all sorts of weird bacteria and viruses and everything that are in the snake's mouth, and he's beginning to look at that connection as well. So yeah, the microbiome, I've always said that's another interest of the shows, mine and the show, but yeah, I think we're only beginning to realize the connections there. So that's exciting that you're thinking about that as well. Yeah, then one other thing I wanted to ask you about, and I read once again in your bio when I was going through all the other stuff that you're looking at, outside of the vision domain and neuroscience, of course, and the one that struck me that I'd like just to ask you to talk a couple minutes about is the coat patterning, and this is while it might not seem patterning dynamics of the coat, when you look at these this organism, and you got the stripes and you have the dots and you have the one, some are black and some are white and all that bit, is really a very unique looking creature. And this whole theme of patterning, not just from, hey, this is what the coat looks like, but patterning in general, whether it's the patterning of how a heart is formed or the brain or any of the complex organs of our bodies, is something that's been a mystery for many years. This whole area of morphology, morphodynamics, and so forth. Can you say a couple words in what you've looked at in terms of the coat patterning work, but any other interesting insights, that might pop, if you shave off the dots always come back in the same place, and and how maybe that could translate to, you see where I'm going with that?
Okay, so the work you're referencing is out of the lab of Ricardo Mallarino, who's at Princeton, and we started working together when he was still postdoc in Hopi Hextra's lab at Harvard. And so that work is ongoing, and there's little that I can say about it at this time. I will say that when an animal's skin is damaged, which is rare in the colony, but I've had thousands of animals over 20 years now, so there have been times when an animal has had damaged skin, that scar tissue, like we think of it, forms, and there is not a recapitulation of coat patterning on that scar tissue, okay? That's number one. Number two is if animals lose their hair from self-plucking, that's a rare but not never scenario that we see in captive animals of all kinds. Animals will pluck the hairs out of their tail. Yeah. We've had animals that rub themselves, absent any parasites or any medical issues, on their tunnels, and they'll lose some hair temporarily. There's also a condition that we've identified. We call it goggle eyes. In the spring, breeding males are under a lot of physiological stress, and they, even if they're not in an environment where they can get to 1 million females in a day and try to, are you in estrus? Can I have it? Can I have it? They're still in stress in the laboratory environment. So in the spring, during breeding season, and if they are housed on a particular kind of bedding, we've had to experiment with beddings because our animals live in a high humidity environment. Their skin is adapted to soil and wet grass, high humidity contact, okay? In the Midwest, it's pretty wet here. Lab bedding is dry. And also if they are fed a ration that is based on corn. They lose the hair around their rims of their eyes, and then that hair loss can actually spread on the face and down the throat. And it's really characteristic. And there's no parasites, there's no infection, it doesn't itch, it doesn't hurt, they just lose the hair. And after breeding season is over, any animals that have it, the hair goes back and the follicles are intact. And so that's just a hair loss issue. I know what you're asking. I will just say this, that Dr. Mallarino's lab is tracing the onset of coat patterning, and our animals are called 13-line ground squirrels, but they have stripes and spots.
Yep.
And, you know, maybe the picture you can be seen, I don't know, but our viewers can look it up. They have stripes and spots, and he's working cross models. It's wonderful comparative Evo-Devo, as it's called.
Yeah, that's where I was going.
Right, and so they're working very carefully, and I believe the work will be published soon on the developmental onset of the genetic programs in our species to compare to others.
Yep.
I think it's the African striped mouse that they've done the most work on, but I know that he works on multiple models, and so I can't talk about the findings, and but it's fascinating stuff. And why does coat patterning matter? In our lab, again, my collaborator in hematology and lacrosse, a few years ago, they've always captured their animals since getting the ones from me in a single golf course in La Crosse, Wisconsin. And they've identified not just wild type coat patterning, but 1, 2, 3 different variants, naturally occurring variants in what's probably an inbred sort of landlocked optimal habitat for a population that's inbreeding. And they've provided us with those animals, and we've bred one line out of it. We call them the blondes, but they have the spots and the stripes, but they're pale, like a Palomino horse, if you're familiar with golden color. And so the pattern is preserved, but the background color is different. And that may or may not be interesting. We've had the genetics worked up on that, and the data are pending because there's a hypopigmentation syndrome in humans that causes eye problems, and we do see some eye problems in our blonde ground squirrels, which is a naturally occurring mutant. Guess what? 4 days ago, a woman who lives a half a mile from me, 3 hours from La Crosse, sent me photos of a blonde in her yard. So this is so there are these genetic variations among wild populations. I found one report in Illinois, photographic evidence of pure albino 13-line ground squirrels, pink eyes, white. And I contacted the homeowner as soon as I found this, and he said, yeah, my neighbor poisoned all of them because he hated them. So so these sports of nature, if you will, do occur. Coat patterning provides animals safety in a background, and our animals are short grass prairie, but they're very visible because of its short grass, and stand up straight on 2 feet a lot to view their environment. And so why are spots and stripes good? Is it species recognition? Because there are overlaps of different ground squirrel species, and there is hybridization. Those have been found and validated through genetic study. I don't know the purpose of them, and I can't talk about the ongoing work, but it will be. Mallarino.
You knew where I was going with that, and I just, I appreciate you just talking about it for a couple of minutes there, but it's just once again the principles of patterning and broader of that of form are just as I said, it always seems like such a it might look like a simple thing, but it is one of those mysteries that it's it's always difficult. It's not just this gene or this protein over here. It's really a very complex process. So I appreciate you at least talking about it from that angle.
Well, and nature tends to conserve mechanisms. So what controls coat patterning may control limb formation in some iteration.
Let
me just say too, that I work at an undergraduate institution. My whole career here on the background of squirrel research, I've taught a full load for all but one sabbatical year, a full load of classes, and our class sizes have gotten very large in the 250 student range. And so teaching and committee work and then research is, I won't say it's a sideline, but we are teaching campus. And since retiring last June, I'm now able to focus more on collaboration.
Yep.
Before, knowing that so many programs were dependent on us producing animals, I became a reproductive biologist, which introduced me to some endocrinology, obviously a lot of behavior. You think about genitalia and their readiness, because our animals don't have ready genitalia 365 days a year. That's been my focus, and it's very practical, but it's borne fruit for all these other things. Yeah. But I got to shout out to all the clients and collaborators that have gotten squirrels from us and included me to the extent I've had the time for. It's been an absolutely magnificent time.
Yeah. Dana, we talked about NIH. You mentioned Fauna Bio, and that announcement was in the press recently. Anything else hot for the rest of 2022? Anything else you want to mention? I mean, give you the floor, but conferences that you may be talking at, any other places we can see you or meet you, or please just take the floor, and anything else that you might want to mention.
Yeah, well, I used to go to the research ARVO, Association for Research in Vision and Ophthalmology meetings. I was a regular at those meetings until 2008. The dates coincide with breeding season in the colony. And then some fall meetings like neuroscience and cell biology I'm teaching. And so I haven't been able to go to meetings since 2008. So sure. People will start seeing me. I'm hoping to attend ARVO next year. The Association for Animal Lab Science, I can't remember the acronym, but ALAS has a really nice meeting that I'd like to go to. It's very germane to husbandry and emerging model organisms. Anyone in the audience who's familiar with CITI programs? CITI is a company that creates training modules for animal care staff. And they've been after me for a while to do a training module for the CITI program for 13-line ground squirrels as an example of a non-traditional species. And I'm hoping to have time to actually get started on that. They've been waiting a couple of years for me. And so that's not a meeting exactly, but it would be something where it's a sign that the model I've bred for so long is mainstream enough that we need training modules for it. And I do. 2 weeks ago, I hired my first ever full-time employee, and maybe followed by more. Once I'm confident I can miss breeding season, I'll be at the meetings. But ARVO and ALAS and maybe cell biology. I don't know. I haven't thought about meetings in so many years. I haven't really figured out a schedule yet, but it's not possible.
Well, anyway, it'll be nice to continue to follow your work and when they meet you out there on the road, because I mean you've started something here with this set of programs, which as we've been chatting, it goes in so many different directions. This cryptobiology, hibernation, physiology, it's just such an untapped territory, and I think you're really leading the path here with what's going to happen next. You got the collaborations happening, and just really going to be rooting you on with everything that continues in terms of the development of this translational possibilities for humans, whether it's human healthcare, whether it's getting us into space, further into space. So really, really exciting work, Dana.
Thank you.
For everybody that is going to be listening to this particular episode across the various podcast networks or watching on the YouTube channel, again, you've been listening to Dr. Dana Merriman, Professor Emerita in Biology, University of Wisconsin-Oshkosh, Director of the Squirrel Colony, and also Adjunct Professor Ophthalmology and Visual Sciences, the Medical College of Wisconsin. Dana, it's really very interesting discussion. I want to thank you for taking the time out of your schedule to come talk about the history here. Obviously, thank you for doing it and setting the groundwork for this future. And as we say on this show, thanks for helping to create a better tomorrow through the work you've been doing. Really very exciting story, and I wish you the best with it.
Thank you.